any antibiotic which is taken can act on the bacteria in the following ways. It can interfere with the protein synthetic mechanism, which majority of the antibiotics like tetracycline, chloramphenicol, aminoglycosides, macrolides, linezolide, everything acts and interferes with the protein synthetic machinery of the bacteria. The second main mechanism of action of a bacteria, it inhibits the nucleic acid synthesis, especially the fluoroquinolone like ciprofloxacin, ofloxacin. It binds with the DNA gyrase or the topoisomerase and interferes with the expansion or formation of a bubble in the DNA or the unwinding of the DNA for either replication or for the transcription process that is inhibited. So the bacteria cannot replicate, it can't transcribe into mRNA. So that is how fluoroquinolone work. That is the second mechanism of action. Inhibition of cell wall synthesis, especially the penicillins and cephalosporins, they interfere with NAG and NAMA. NAG means N-acetyl glucosamine, NAMA means N-acetyl muraminic acid. There is a bond formation between these two molecules in the cell wall of the bacteria. So when penicillins and cephalosporins are given to the patient, it interferes with the bond formation between NAG and NAMA. Therefore, there is no bond. Therefore, the cell wall becomes weak and it becomes porous. By osmosis, the liquid will enter the cell, bacterial cell, and the cell will become fluffy or puffy and it will ultimately rupture. There is the penicillins and cephalosporins or beta-lactam group of antibiotics work by inhibiting the cell wall synthesis. Disruption in the membrane function, that is the cell membrane, will undergo lysis. Therefore, it is somewhat similar to penicillin group of antibiotics, but it interferes with mainly the lipid bilayer of the cell membrane or the lipid bilayer of the cell wall component, inner layer of the bacterial cell wall. The folic acid interference in the metabolic pathway, that is the last mechanism of action. The sulfonamide group of drug which utilizes this pathway, there is something called tetrahydrofolic acid even in the bacteria for metabolism purpose. This tetrahydrofolic acid is formed by the dihydrofolic acid or the folate. The ingredient for this is PABA, para-aminobenzoic acid. So when you interfere in the formation of this folate or the folic acid by blocking the PABA or the reductase group of enzymes, so thereby interfering in metabolism, so no energy or no tetrahydrofolic acid active form of the folate. So therefore the bacteria is going to die due to lack of metabolism. So in summary, the antibiotics works by following mechanism: by interfering with protein synthesis, that is majority of the antibiotics works in that mechanism; inhibiting the nucleic acid synthesis within the DNA of the bacteria; inhibition of cell wall synthesis, that is in the cell wall of the bacteria; disruption of the cell membrane function by causing a pore, extra hole will be there and leakage of all the cytoplasm will be there in the bacteria; or interfering with the metabolism, especially the tetrahydrofolic acid synthetic pathway. It is usually sulfonamide group of antibiotic. Thank you for watching and learning from Logic Medico. If you are new to our channel, kindly consider subscribing to our channel. Like this video and share this video with other friends. Hope it helps. Thank you once again. Good day.